Hey there everyone, and welcome back for more biotechnology. So let's go ahead and continue our discussion on studying proteins. So the focus of this video is going to be on how a cell actually goes about making proteins, some details of which we have already covered before, but the goal here today is to get a little bit of a more complete understanding about how a cell actually goes about doing this so that we can understand moving forward in a biotech or laboratory setting how proteins operate and how we can actually go about making proteins for some particular purpose, whether it's academic or for profit. So protein synthesis is something that a cell is basically going to be doing all the time. If a cell isn't making proteins, it is not going to be living for very long. I always like to say that proteins are what actually does all the heavy lifting in the cell. The DNA is just the blueprints on how to make the proteins that a cell needs in order to survive. And for a biotechnology or laboratory setting, we need to understand how a cell actually makes proteins because in most cases, that's going to be the only way we can get our hands on proteins is to have a cell make the protein and then we find a way to harvest it. So we've already discussed this as it pertains to the central dogma of molecular biology, which you'll recall says that we make proteins in a two-step process. We take the DNA, we pick a gene, we transcribe that into a messenger RNA molecule, and then we take that messenger RNA molecule and translate it into a protein. So if we break this down on a step-by-step basis, as we just described, the transcription part of this, in which a single gene on a chromosome is turned into a messenger RNA transcript by an enzyme that we've already mentioned called RNA polymerase. So essentially what we're doing here is we are making a copy of the gene because the gene isn't going to go with us. So the gene has to stay as a part of the chromosome inside the nucleus. So we're basically just, for all intents and purposes, we're making kind of a photocopy of it that we can take with us and we can still read and get all the information we need. So there is some messenger RNA processing that often does occur. We haven't really ever mentioned this before, but there are little sequences within the messenger RNA that may actually not be necessary for synthesizing the protein, and these are called introns. So there are enzymes within the nucleus that can snip these introns out in cases in which they are not needed, and we can splice together the messenger RNA so that it only contains the parts that actually code for the protein. So once the messenger RNA has been processed, we are going to export it out of the nucleus so that it gets into the cytoplasm, which is where the ribosomes are located. So once the messenger RNA is clear of the nucleus, it will bind to a ribosome, and the ribosome is essentially your construction crew that is going to read the instructions in the messenger RNA and use those instructions to start using the raw materials in making a protein. So the signal that the ribosome is looking for on that messenger RNA transcript is the start codon, because that basically says this is the start of the instructions, this is where you start building the protein. Everything that comes before the start codon is essentially ignored. So once that start codon is encountered, the ribosome will begin assembling the polypeptide chain, what is going to become the protein, by utilizing a new type of RNA molecule that we have not discussed yet called transfer RNA. Transfer RNA is basically the adapter between the nucleotides within the messenger RNA and the amino acid that we are going to use to incorporate into the protein. So tRNA molecules are each attached to a particular amino acid, and they are going to come into the ribosome, deliver that amino acid depending on which codon within the messenger RNA is encountered. More on this a little bit later. Okay, so let's talk a little bit more specifically about the transcription process. All this is we are taking the gene that contains the DNA instructions on how to make the protein, and we are rewriting those instructions into an RNA molecule. So the gene is incorporated into the DNA, but like I said, we can't really take it with us. So the whole transcription process is necessary for a couple of different reasons. So like I was saying, the chromosome is usually going to be very, very large. We're usually just picking one gene at a time, but the whole chromosome is going to contain possibly thousands of different genes, and maybe we don't need to transcribe the other 999 of them, right? So if we only need to transcribe one particular gene, it wouldn't really make sense to just take the whole chromosome with us. So if you go to the library and you find a really big and nice cookbook that you just want one specific recipe from, it might make more sense to just make a copy of the recipe that you want rather than taking the whole cookbook with you, right? So number two, even if we did want to take the whole chromosome with us, being that the chromosome is so bulky, we're not really going to be able to take it out of the nucleus to where the ribosomes are. It's a problem of the construction crew and the blueprints not being in the same location. So imagine that you've got blueprints for building a house, but you can't take those blueprints out of the library or wherever they're contained. And the construction crew can't come and build inside the library because that's not where the house is supposed to be built. So by using transcription, we can get a copy of the blueprints to the construction crew, even though the original blueprints are never going to really leave where they originate. And then finally, the ribosome and the tRNAs that are bringing the amino acids in for us to use, they are specific for RNA, so they can't really recognize a DNA code. Specifically, the thymine nucleotides that are in the DNA, the ribosome and the transfer RNAs, they're not really going to know what to do with that. They speak a slightly different language in which we use uracil instead of thymine. So for all these reasons here, we need this transcription process. But once we actually do make our messenger RNA transcript, it should be perfectly complementary to the gene's DNA sequence, just with those thymines replaced with uracil. Otherwise, the information should all be exactly the same. We should not have made any errors whatsoever in the instructions for how to make our protein. So speaking of making our protein, that is what the translation process is all about. We have our messenger RNA molecule, it's been processed, it has been exported out of the nucleus, and the ribosome is ready to bind to it, read those instructions, and start making our protein here. So these ribosomes can exist in one of two different places. They are either freely floating around in the cytoplasm, or they are embedded within the rough endoplasmic reticulum. So regardless, the process is going to work largely the same way. The translation process is going to involve the ribosome reading the nucleotides in the messenger RNA transcript in sets of 3, and these sets of 3 nucleotides we already defined as codons. So a codon is a stretch of three nucleotides within the messenger RNA transcript that are each going to code for one particular amino acid. And we talked about that before when we talked about the genetic code, and we're going to see that again here in just a minute. So once we encounter that start codon, which is AUG, every codon after that is each going to code for a particular amino acid, so that every time we go from codon to codon to codon, we stick another amino acid and another and another and another onto a growing polypeptide chain. So you can actually see that as the ribosome scans throughout the codons in the messenger RNA transcript, it starts sticking amino acids onto each other by forming peptide bonds. So what is a ribosome exactly? We've only really described it as this kind of protein-building machine up until this point. So the ribosome is a very large multi-subunit hybrid molecule. So it consists of both protein components and RNA components. The RNA components are called rRNA, or ribosomal RNA. So when these components join together, it basically forms this big machine that is capable of binding to the messenger RNA, and it also has enzymatic activity that allows it to form bonds between adjacent amino acids, and that's going to be how we actually make a very long polypeptide chain that has many, many amino acids incorporated into it. So the transfer RNAs, which you can see here, being RNA molecules, they are made up of nucleotides as well, and they contain regions of their own nucleotides called anticodons that are going to be complementary to the codons found in the messenger RNA. So depending on the sequence of the anticodon in the transfer RNA, that will determine which amino acid the transfer RNA becomes charged with. So if we look at the ribosome here, the ribosome actually contains 3 little cavities or pockets in which transfer RNA amino acid complexes can occupy. These cavities are labeled EPA. I like to remember that as the acronym for Environmental Protection Agency, EPA. It's kind of easy to remember. So what these letters actually stand for are, the A stands for the entry or access site, so that's going to be where the first tRNA amino acid complex enters. The P in the middle stands for peptide bond forming site, and the E stands for the exit site. So as the picture is drawn here, tRNA amino acid complex are going to enter the ribosome from the right and then exit through the left as they move from right to left there. So the first tRNA amino acid complex will enter the A site, and it will be one in which the anticodon is complementary to the start codon, which is AUG. And this tRNA is always going to be attached to the same amino acid, which is methionine. You will recall from the genetic code, which we will see a little bit later on, that the start codon AUG always corresponds to the amino acid methionine. So that is the reason why many proteins always begin with the amino acid methionine at their N-terminus. So at this point, the ribosome is going to continue scanning down the messenger RNA from 5' to 3' direction. So as that happens, that first tRNA is going to be moved from the A site into the P site. And then at the same time, the next tRNA will enter the A site adjacent, so we now have a situation in which there are tRNA amino acid complexes that occupy both the P site and the A site. So at this point, those 2 amino acids, the methionine in the P site and whichever amino acid is in the A site, they're in very close proximity to each other. So now the ribosome is going to use its peptidyl transferase enzyme activity within the ribosomal RNA to forge a peptide bond in between those 2 amino acids. So that's going to be a peptide bond formed between methionine and whichever amino acid comes next. So the methionine is now going to dissociate itself from its tRNA that now that it's joined to another amino acid, and that particular tRNA is then going to move into the E site, and it is then going to exit out of the ribosome. And the second tRNA that was in the A site is then going to migrate over to the P site, making room for the third tRNA molecule here to now occupy the A site. So this is basically just like an assembly line in which the tRNA that we're done with exits out and a new tRNA comes in. So this process of manufacturing proteins is going to continue as more tRNA amino acid complexes are brought in that each have codons, that each have anticodons that are complementary to what comes next in the messenger RNA transcript. So as we continue to do this again and again and again, we form more and more and more peptide bonds between amino acids in the A and the P site. The polypeptide chain is going to continue to grow and grow and grow as more amino acids are attached to this growing chain by the ribosome itself. So eventually, as the ribosome scans throughout the messenger RNA, it is eventually going to encounter a codon in which there is no anticodon on a tRNA molecule that is complementary to that particular codon. So these are called stop codons, and there are 3 of them: UAA, UGA, and UAG. So because there are no tRNA anticodons that correspond to these particular codons, once the ribosome encounters one of these codons, the protein synthesis is going to stop. So this is why we call it the stop codon. Once we encounter one of these, the protein synthesis stops, and the polypeptide, as it has been made up until this point, is going to be released, and now we can start worrying about protein folding, as we discussed in the first video of this chapter. So here is another look at the genetic code, which of course we have talked about before. You certainly do not need to memorize this at all, but what you should probably remember is the sequence for the start codon and the sequences for the stop codons. Everything else you can look up as necessary. So you will see the start codon, which corresponds to methionine here, AUG, and then the stop codons UAA, UAG, and UGA. Everything else, every other codon, will code for a particular amino acid. And since there are a total of 64 different codons and only 20 amino acids to code for, you can imagine that there's going to be some redundancies. So for example, there are 4 codons that all code for glycine, 4 codons that all code for valine, 4 codons that all code for alanine, threonine, proline, serine, and so on. So it's not necessarily true that one codon only codes for one particular amino acid. There are going to be some redundancies there. So based on this, we've gotten some practice with this before, but based on this, if your messenger RNA reads from 5' to 3', UAU, AUG, UUU, CCG, GCA, and UAG, then the translated sequence of amino acids here would be from N-terminus to C-terminus, methionine, phenylalanine, proline, and alanine. Well, your first thought might be, well, what about this UAU? That doesn't code for methionine, so if we actually look it up here, that codes for, if our first base is U, our second base is A, that would code for tyrosine. So why is tyrosine not here? Well, you have to remember, we don't start building the protein until we encounter the start codon, right? So this first codon that you see here is ignored because we have not encountered the start codon yet. Here is the start codon right here, so that's why the first amino acid incorporated from N-terminus to C-terminus will be methionine. And then after that, UUU stands for phenylalanine, CCG stands for proline, GCA stands for alanine, and then we don't add any beyond that because UAG is one of our 3 stop codons. So like we said, the start codon has to be encountered by the ribosome before polypeptide synthesis can begin. And then once one of those 3 stop codons is encountered, no further codons are going to be read at that point. And then your book does spend just a second talking about bacterial protein synthesis. The nice thing, though, is that this is largely going to be the same. All the same machinery is there. We still are using messenger RNA, tRNA, and ribosomes, so there's not a whole lot of difference here for us to really get into, other than to say that we're dealing with a circular chromosome in which the DNA is on. There's no nucleus, and there's no rough endoplasmic reticulum. Otherwise, everything is similar enough. So a couple other things to say, that once a protein has actually been fully synthesized and released from the ribosome, it's going to fold into its final tertiary configuration that we have talked about before. We've said that a protein has to be folded into its particular 3-dimensional shape in order for it to have any function at all. But even after this has occurred, the protein can be further modified, and we call these types of modifications, after translation has been completed, post-translational modifications, or PTMs. So depending on which type of post-translational modification a protein receives, this can actually greatly alter its function or its activity. The best example of this is what is called phosphorylation of a protein, and this type of post-translational modification is performed by a type of enzyme called a kinase. So what kinases do is that they essentially take a phosphate group off of an ATP molecule, which you'll recall is our big energy molecule inside the cell, and it takes that phosphate and essentially attaches it to the side chains of certain amino acids within the protein. So essentially we are tagging the protein with that phosphate group. Phosphorylation can act an awful lot like a switch, like an on and off switch. So depending on whether a protein is phosphorylated or not, that protein can either be completely switched off, in which it has no or little activity, or it can be completely switched on, in which it has a great deal of activity. So phosphorylation can be a very powerful way for cells to switch proteins on and off in cases in which they need them or they do not need them. Another one that your book talks about is called glycosylation, which is the attachment of sugars onto proteins to form what are called glycoproteins. So glycoproteins are very commonly found on the surface of the plasma membranes of cells. And these glycoproteins will often function in cell recognition capacities. For example, blood typing. Depending on what type of blood type you have, A, B, or O, your red blood cells are going to be covered with certain types of glycoproteins, and the immune system is going to recognize different types of red blood cells depending on what types of glycoproteins are there. And your blood type essentially tells you which types of glycoproteins you have on the surface of your red blood cells. And then another type of post-translational modification. There are actually many types of PTMs that we could talk about, but one of the major ones to discuss is called cleavage. So this is not actually a case of attaching something to a fully translated protein. This is actually a case of cutting a protein into smaller fragments. So cleavage is going to be mediated by a type of enzyme called a protease. A protease is an enzyme that breaks peptide bonds between amino acids in a polypeptide, kind of like a pair of scissors. So those smaller fragments of proteins can then go on to act independently of one another. So oftentimes what a cell will do in the body is it will synthesize a very large inactive protein, and then snipping it into a smaller protein will actually activate that protein and allow it to have whatever its intended function is meant to be. So as I said, these are certainly not the only types of post-translational modifications, but this should just kind of give you an idea of what these are like and what they tend to do. Alright, so that is going to do it for this particular video on protein synthesis and how proteins are made. So join us next time and we will start talking about a particular and very important class of proteins called enzymes. So I look forward to seeing you next time. See you then.